microscope and your box of slides, or you can look at what I'm projecting and then compare it to your diagram on page 246 in the lab manual.
Which slide is that?
Human bone, slide 52 out of the box of slides.
So, like, on this.
Did I get the number right? Yeah, compact bone, yeah, 52. But it does come from a human being. But it wouldn't matter if it were from some other vertebrate. Now, what I hope will catch your attention about this bone tissue slide is that bone tissue is not composed of a bunch of cells like you have been looking at previously at the epithelial tissue and the muscle tissue and whatever other tissues you've looked at. There are no cells for you to see on the bone tissue slide because they're not there. Now, when this tissue was alive, there were bone cells present, but that slide is very old and the cells have deteriorated. They're gone. I will be able to show you where they were, but they aren't there anymore. But what you can see is that bone tissue is made up of repeating circles of bone matrix. And each of these circular regions is called a haversian system. In the center of a haversian system is an opening. Now, depending on shading, it's either going to appear light or it's going to appear dark. But it's a hole. And this hole was occupied in the living tissue by an artery, a vein, and a nerve. All right? But those structures also are no longer present. The bone cells were, notice that's past tense, were located in these dark objects. Now let me show it to you on this chart because it's a little clearer than that display. These dark rectangular objects are not structures, but they're holes in the bone matrix. And that's where the bone cells were located when this tissue was alive. But the cells are not present. Now, the function of these bone cells, like we just finished in lecture, is to secrete bone matrix. Now, on this chart, and I know it's a diagram, all of the pale yellow material is bone matrix. And like I talked about in lecture just now, that bone matrix is hard. So you might want to think of this tabletop as being like bone matrix. Very dense, very hard, and rigid. Okay? So you are asked to know the bone matrix. You're asked to know the holes where the bone cells were located. Now those holes are called, here's the singular form of the word. One of them would be a lacuna, but more than one would be lacunae. All right. Am I correct that these structures are labeled on page 246 of your lab manual? Are these parts of the bone matrix, bone tissue labeled? Can somebody confirm that for me, that it is there? Okay. Now, and I've told you about the haversian canal, so we're down to this last structure called the canaliculi. Now, there's not one of them that's the plural form of the word. All of these, what look like black lines on this diagram, or if you look up here, you can see the canaliculi pretty easily there, as well as elsewhere. Here's some. These are grooves in the bone matrix, where part of the bone cell was lying. So over on this chart, and this is very helpful, I think. You can see that the bone cells have that oval shape just like the lacuna, but these structures that project off of the bone, major part of the cell body, are kind of like the pseudopods of an amoeba, because this is cytoplasm with the surrounding cell membrane. All right? And every one of these structures would be a canaliculus, that's the singular form of the term, and all of them are referred to as canaliculi. All of these narrow black lines that you see here, or here, or here, or here, are the canaliculi. Other questions? Be sure you make a notation. You are responsible for the bone tissue slide for lab exam 3. This is week 11 of the semester. That exam will be 4 weeks from right now, on Thursday of week 15. Yes, ma'am.
The osteocyte? Does that sound, am I saying that? I might
All
not
right,
be
the
saying it
term
right.
osteocyte is a term for a bone cell. Now, the osteocytes are mature bone cells. They are not, they don't have the same function as the osteoblasts or the osteoclasts. And we were just about to get to that in lecture, the difference between those different kinds of bone cells.
So on there, an osteocyte with lacuna would be...
I'll be
like
honest
just
with you, she cannot ask you an osteocyte because there's none on the slide.
So it would be where that like empty circle would
But
be.
they were in those lacunae.
So in the white spaces?
No, the dark ones.
Oh, the dark black spaces. Okay.
Yeah. But she cannot ask you to identify an osteocyte because it's not there. She might ask you what occupied this space. If she asks a question like that, tell her she shouldn't. She'll like that. All right? That's the only slide you have to deal with pertaining to the human skeleton. Before I forget about it, make yourself a notation that for next Tuesday, that's the 14th, you need your dissecting instruments. And if you are squeamish about touching a brain or the eye of an animal, then bring your gloves. Otherwise, you'll be doing it with your bare skin.
Is the whole thing of the matrix?
This whole circular object is one aversion system. Okay? And then, was that your question?
Bone matrix.
Bone matrix. Bone matrix is everything that you see here except for this hole and those black things, whether they're the lines or the ovals. Now I want you to think of a bone, okay? Do you imagine that this bone tissue, let's just take this humerus right here, that's carved material, isn't it? Very dense. But is it porous? Is this tissue porous?
Yes.
Meaning, does it have a lot of holes in it? Yes. Okay? So this is bone matrix. But if we look at it microscopically, we see that there's a bunch of holes in it and little channels. Okay, are we okay on bone matrix? On this chart, here's the aversion system. Everything that's yellow is bone matrix. Okay, about Tuesday now, make a notation because You have to have these dissecting instruments, and if you don't want to touch it with your bare fingers, have your gloves. All right, now, some other aspects of the human skeleton that you are responsible for is in the lower left corner of the screen here. Let me turn this off. Over here on the cabinetry, there's a stereoscope. And on this stereoscope, I have 3 bones that are very small that are present in your middle ear. Those ear bones collectively are called ossicles. The term is up there. But the 3 bones are so distinctive in their shape that they have been named Malleus, Incus, and Stapes. You perhaps know them better by their common names, hammer, anvil, and stirrup. Now these 3 bones are attached to each other so that when sound waves cause your eardrum to vibrate, the vibrations are transmitted through these bones to your inner ear. And only if that transmission occurs will you hear a sound. All right? Now, they're big enough where you can see them with the naked eye, but I've got this magnified 80 times, 80. And with that magnification, that just makes them a little bit easier to see. They have been removed from a cadaver and glued into the bottom of this box. Be sure you look at that. Then the next thing pertaining to the skeleton that I want to call to your attention is on the front table here. There are some bones in the skeleton that are not connected to other bones by ligaments. They're called sesamoid bones. I wrote the word on the board there. And in the human body, we have 2 sesamoid bones. One is the patella, better known as what?
Your kneecap.
The kneecap. And the other one is called the hyoid bone. Now you know where your kneecap's located because you can feel it. And I've got one up here. And this is a human patella. This is not plastic. It came from a skeleton such as that one over there. But what you have probably noticed about your kneecap is if you grasp it, you can move it. It's not held in a rigid position. A second sesamoid bone that's present in the human body is called the hyoid bone. Here's a real one. But it is present over on this skeleton, and you'll find it located under the mandible, right here. Now, they've attached it to the skeleton with wires because they had to have some way to hold it up here. But it is not attached to other bones by means of ligaments. What do you think might be attached to this in your body?
Your tongue.
Your tongue?
Your tongue. Okay. Tongue is muscular, and like we just talked about in lecture, in order to move body structures, you got to have muscle tissue to move it, but there has to be an anchor for that muscle, and that anchor is the hyoid bone. Now, in other mammals, not human beings, there's another type of sesamoid bone that's located within the penis, and that bone is called a baculum. Rodents have it, and so I have here it is, an example of a rodent's baculum. This is not plastic, this is the real thing. This is from a mink. Minks are rodents. This is the baculum of a raccoon. This is the baculum of a coyote. And of course, what's the purpose of the baculum? so that the penis will be erect. All right? And this is the baculum of a walrus. This is a dude. Now, this particular one is partially petrified, so when you pick it up, you'll notice it's extra heavy, heavier than bone. It's because some of the bone tissue has been replaced by minerals when it was buried somewhere in the soil, and so it's become petrified. But the Alaskan natives actually use the baculum of a walrus as a tool. As you can see, it would make a very good club, and they use that to subdue their prey, which is a variety of animals. I assume what they do is they crush the skull with this. All right. Then there's a couple of things I need to tell you about these bones that you're going to study. Over in these boxes on the cabinet that look kind of like suitcases, we have a variety of human bones. Some of them are real, and some of them are made of plastic. Here's a plastic femur, a right femur. But when you look at it closely, you'll see it has lots of irregularities on it, and you wonder, why did they just do a sloppy job of making this? No, this is a cast of a real bone, so that it will look like a normal bone, even though it's plastic. It's almost impossible to obtain real bones any longer. This skeleton, I can't imagine how many decades old it is. I know personally that it's more than 40 years old. And I don't mean the age of the person whose bones that belongs to. I mean how long this college has had this skeleton. But the cost of real bones is just off the chart and beyond our capability to spin. So you'll find a variety of bones over here. Here's the bones of a complete foot. It's my understanding from Mrs. Hughes that if she were to ask you some bones from a hand or a foot, that she would put out the complete foot or hand and then ask you, you know, what's the name of this bone or whatever one she chose, instead of disassembling the foot and then asking you those names. What else? Oh, I want to say this about studying the bones. Most of you, and I'm no different, would feel compelled to touch the bone that I'm studying, or the part of the bone that I'm studying. I'm asking you, please do not touch it with any writing instrument. The real bones are the bones of a once living person. I think we should have that much respect for that person that we don't mark up the bones with pens or pencils. If you need something to point out parts to yourself or your lab partner, in this beaker here on the front desk there are some probes. If you have your own, of course, you're welcome to use that, but please do not use any pen or pencil. Then I got a couple of specimens I want to call to your attention. This being one of them. This is a fetal skull, and this one is plastic. This is not real, but it's a cast. And when you look at the fetal skull, of course, it's not as big as the adult skull, but if you look at the facial features, you see that they are not as developed as the rest of the skull is. The major part of the skull is more developed because the brain has to start development early. As you're going to learn when you cover embryology, that's still coming. The brain development has to get started very early after fertilization. Now, as far as the bones of the skull, on your skull, the bones like the big parietal and the occipital at the back and the frontal bone at the front, they're all fused or grown together. But on a fetal skull, that's not the case. The the bones have to be able to shift in relationship to each other in order for